Hi everyone, my name is Hoyoung Lee. I'm a graduate student working at Professor Uichul Shin's lab at KAIST Daejeon, South Korea. Today I'm going to talk to you about our recent work regarding the characteristics of IL-15-induced bystander-activated memory CD8 T cells in viral hepatitis. I'll start by giving you a brief background of this presenting work. IL-15 has been recognized as a key cytokine required for homeostatic maintenance of memory CD8 T cells. But our previous study has demonstrated the role of IL-15 in inducing bystander activation of memory CD8 T cells that contributes to liver damage during acute hepatitis A infection. In our previous study, IL-15 was shown to activate pre-existing memory CD8 T cells that are not specific for disease-causing virus such as HAV. These bystander-activated memory CD8 T cells were expressing increased level of NKG2D and exerted NKG2D dependent innate-like cytotoxicity, which was associated with severe liver damage in patients with acute HAV infection. Interestingly, expression of NKG2D was only induced by IL-15 in the absence of TCR stimulation. So this indicates the expression of NKG2D is selectively increased in bystander-activated memory CD8 T cells. In the current study, we were able to find the expression of CCR5 was increased by IL-15 stimulation among various chemokine receptors. And in a manner similar to NKG2D expression in our previous study, while TCR stimulation had no effect on the expression of CCR5, it rather inhibited IL-15 induced expression of CCR5. So this increased expression of CCR5 was shown to mediate migration of memory CD8 T cells that are activated by IL-15. We stimulated sorted memory CD8 T cells with IL-15 and placed them on the upper inserts in transwell system and placed CCL5 in the lower chambers. Following 3 hours of incubation, we found CCL5-induced migration was significantly high in IL-15 treated memory CD8 T cells. Next, we adaptively transferred OT1 T cells into naive B6 mice and primed these transferred cells with vaccinia virus expressing ovalbumin. Subsequently, we isolated prime OT1 T cells using a congenning marker, then stimulate them in vitro with IL-15. Following in vitro stimulation, we found increased frequency of CCR5 expressing OT1 T cells and a pattern similar to TCR stimulation that I described in the previous figure, stimulating OT1 T cells with OT1 cognate peptide called Sympacil significantly abrogated IL-15-induced expression of CCR5. Next, we performed in vivo migration assay by introducing PR8 infection in mice that received OT1 T cells that are stimulated in vitro with IL-15. And we treated these mice with IL-15 with or without CCR5 antagonist called maraviroc. As a result, we found that migration of IL-15 treated OT1 T cell was significantly reduced by maraviroc treatment in the lungs of mice infected with PR8 infection. We also observed decreased IL-15 induced expression of Ki67 following maraviroc treatment. We next analyzed the role of CCR5 on memory CD8 T cells during acute HAV infection. Here we found increased frequency of CCR5 expressing memory CD8 T cells in patients with acute HAV infection. We also confirmed increased expression of CCR5 on bystander-activated memory CD8 T cells by detecting its elevated expression on CMV-specific CD8 T cells compared to HAV-specific CD8 T cells. And again, through in vitro transwell migration assay, we found that memory CD8 T cells from acute hepatitis A patients migrated more efficiently compared to those from healthy donors. We also found that CCR5 expressing memory CD8 T cells are enriched in the liver of acute hepatitis A patients. And importantly, we show that the percentage of CCR5 expressing memory CD8 T cells from peripheral blood from the patient is significantly correlated with the severity of liver damage indicated by serum ALT level. Finally, to analyze a unique gene expression of bystander-activated memory CD8 T cells, we performed RNA sequencing of sorted memory CD8 T cells stimulated in vitro with IL-15 with or without TCR stimulation. Through a Venn diagram analysis, we were able to identify overlapping and individually upregulated differentially expressed genes from each condition. And when we present them as a heat map, We were able to isolate cluster 1 as a gene set of IL-15-induced bystander activation in memory CD8 T cells. So this gene set was enriched with genes that were associated with NK cell-mediated cytotoxicity and genes related to interferon response. So this bystander activated gene set is expected to not only identify bystander activation of memory CD8 T cells in other viral infections, including viral hepatitis, but also to investigate regulatory mechanisms underlying bystander T cell activation. That's it for today, and thank you for listening.